Hello everyone, this is Varsha Bajpai, science teacher, G.D.Goenka School, Indrapuram. So today I'm here to teach you a very important topic, antibody structure. This topic is quite important from exam point of view also, and these days due to COVID, you all should know about the structure of antibodies, which is actually helping you to fight against the diseases, right? So we will explore this structure of the antibody in bit detail. So let's start, let's continue. This is your antibody molecule. This is your single antibody molecule, which is glycoprotein in its chemical nature, right? Antibody is a glycoprotein chemically, right? That means glyco word is basically taken from carbohydrate part, and protein, we all are aware of that, nitrogenous compound protein, right? So this Y-shaped molecule is known as antibody, and it has two chains. You can see this green region, right? A complete Y. This is the heavy region. This region is known as heavy region of the antibody or heavy chain of the antibody, right? And this blue part is known as light chain or light region of the antibody, right? So this green chain is known as heavy chain, and this blue chain is known as light chain. So we can see total 4 peptide chains are here, right? So that means antibody is a tetrapeptide molecule. Antibody is a tetrapeptide molecules. Now, if I talk about different regions in this antibody, you can see different many things are written here, CH, VH, and you must be thinking, what does it mean? Because when students look at its diagram, they feel like it's very complicated, but no, not at all. I can assure you after this video, you will be clear with the diagram of antibody. Right? So let's start from here. First of all, this VH and VL, what does it mean? It means the variable region or variable domain of the heavy chain. And in the same way, this VL means variable region or variable domain of light chain. What domains mean? What this domain word means? See, domain is just an amino acid sequence in a peptide chain. And we know that antibody is a peptide chain. It's a peptide molecule, right? So whatever amino acid sequences are present in it, they are known as domains, right? So here we will not use the word region. Instead of that, we will use the word domain. It's quite perfect in terms of biology, right? So this variable domain of heavy chain and this variable domain of light chain together constituting the fragment and the region where they can bind the antigen. I will explain you this in more detail, right? Right now we are just having the overview of the antibody. This region is actually the antigen binding site or antigen binding region. That means whenever pathogen enters in your body, right, and your body going to produce immune response and your antibodies are going to attack the antigen. So which is that region, or what is the exact region in the antibody where this antigen gets bind, or the region from where they grab the antigen, right? So that antigen can't run away. So this is the region in between the variable domain of heavy chain and light chain. Now, this CH and Cl means constant region. I will explain you why we call it variable, why this region is known as constant, right? So this CH and Cl means constant domain or constant region of the heavy chain, and constant region or constant domain of light chain. Constant, something which is constant, which does not change, right? Variable which keeps changing. We know that antibodies are specific to the specific antigen, right? And we also know that one pathogen or one microbe cannot have a single antigen. One microbe may have 2 or more than 2 antigens on its surface, and structure of the antigens may vary. So antibodies have to change their structures. Okay, basically this region only, which we are calling variable. This region has to change its structure according to the antigen structure. This is why this region is variable, and we call it variable region or variable domain of heavy chain and light chain, right? So what is important thing to note here, that antibodies are specific to the antigens, and one microbe, a single microbe may have multiple antigens of different shapes. So that is why these regions of the antibodies keeps changing depending upon the antigen shape which they are grabbing it. Okay, now you can see this light chain is very much short. Okay, that is the reason why we call it a light chain, and heavy chain is long, right? Now, variable regions or variable domains are one only, but constant regions are many, constant domains are many here. Constant region 1, constant region 2, constant region 3, okay? But in variable, it is only one, right? Constant region of light chain, constant region of heavy chain. This CH1, CH2, CH3, this is how we denote it. Now, this is the hinge region. We can say it a neck point or neck region. So this is the hinge region from where antibody breaks, right? Antibody cleavage takes place from this region only. That also I'm going to explain you in detail how does it break, and right. So these two chains, light chain and heavy chains, held together by disulphide bond. Okay, how these two chains are held together by disulphide bond. That means two sulfur bonds are binding these two peptide chains together. Now, question is, how many disulphide bonds are there in a single antibody molecules? Basically, 16 disulphide bonds are present in a single antibody molecule, which is holding together two peptide chains. Okay, but 16 molecules, 16 disulphide bonds. These are the combination of the interchain and intrachain bonds. Okay, interchain, that means the bond which is present between these two domains. Okay, and intra, that means which is present between these two chains. Right. Now you can see this labeling here, FAB region and FC region is written here, right? What does it mean? It simply means fragment of antibody region, fragment of antigen binding region and fragment of crystallization region. So what does it mean, and how we concluded that yes, this is the region which is going to bind with the antigen, and what is the role of this antigen? What is the significance of this region here, right? So for this, I'm sharing you one detailed antibody diagram here. Yeah, this is the one. Right. So I'm going to explain the same diagram in more details. So please listen carefully, pay more attention, because it's quite important topic, right? So before that, let me tell you one thing. When researchers were doing research, they took a vessel, right? And they put the antibodies in that vessel. Now, they added one enzyme which is known as Papin enzyme. Papin enzyme, it is written here also, Papin. Okay. So this Papin enzyme is basically used to break the antibody. Okay. It helps to cleave the antibodies. So hinge region, this hinge region is the cleavage site of the antibodies. That means if the Papin is going to break the antibodies, this is the region where it will break it. So if Papin is going to cleave or going to break this antibody, so in how many fragments it will be divided? It will be divided into 3 fragments. 1, 2, and 3, right? 1 fragment, this is the second fragment, and this will be the third fragment. This is important to understand when Papin is going to divide or Papin is going to break the antibodies. So which is the region where is going to cleave the antibodies? It is the hinge region, and then antibodies will be divided into 3 fragments, right? Now, what was the use of this experiment? The use was that when antibodies were break, so scientists observed that after that they added the antigen in that vessel, and they could observe that that antigen is binding in this region. Okay, that means antibodies, even after cleavage, they are able to bind with the antigens, right? So they observed that this is the region which is binding with the antigen, right? I hope I'm clear up to this. Okay, so they said that this is the region, or this is the fragment of antigen binding. That's why we call it FAB. That means the fragment of antigen binding, right? Now, what about this lower region? Being higher in density, this settled on the bottom of the vessel. This region settled on the bottom of the vessel, okay, and got crystallized. That's why we call it fragment of crystallization, okay? Fragment of crystallization. There are many other reasons why we call it FC fragment. Okay. See, we all know that a peptide chain has 2 ends, one amino end and another end is of carboxylic group, right? So amino end is this. This will be the amino end, right? And this will be the carboxylic group end, that is COO, right? You must have learned in chemistry as well. So this region, for this region also we call it FC region. And another important region why we call it FC fragment or FC region, it helps in binding with the complement system. Now you must be thinking, what is this complement system? Complement system is this group of some specialized protein, some 20 to 30 specialized proteins together constitute the complement system, which actually enhance the function of the antibodies. It enhances the ability of the antibodies to kill any pathogens, right? And also it acts as a bridge between innate immunity and adaptive immunity. That means, again I'm repeating, this complement system when bind with this region, right, it gives some signals to the mast cell and the macrophages cells, right? You must have learned about mast cell and macrophage cells. They're the phagocytic cells. That means they engulf the bacteria or virus or any pathogen, and they break the cell membrane and totally destroy the pathogen, right? So we can say this complement system is actually acting as a bridge between innate immunity and adaptive immunity. Innate immunity, macrophages and mast cells are the part of innate immunity, and your B lymphocytes and T lymphocytes, these antibodies are the part of your adaptive immunity, right? So complement system is helping in binding the mast cell and the macrophages with this region of the antibodies. This is how it acts as bridge between the innate and the adaptive immunity. Now to bind with this antibody, these mast cell and macrophages have some receptors on their surface through which they bind with this region. Okay, I will not go in much detail. Okay, you will learn that in higher classes. This is quite enough for you all. Right. So till now we are clear with this variable domain of light chain, right? Variable domain of heavy chain. Constant regions or constant domains are those which are not variable. They do not, their amino acids are specific. Okay. They do not change when antigen comes, right? But these variable regions change depending upon the shape of the antigens, because we know that antibodies are specific to the antigens, right? And this FC region, why we call it FC region? Because this is a region which helps in binding of the complement system, and this complement system enhances the ability of the antibodies to kill any pathogen, right? And now, yeah, one more thing I need to tell here. What is this hypervariable region means? See, these variable regions have inside of them hypervariable regions fitted in them, right? So what is the use of these hypervariable regions? These hypervariable regions are present inside the variable regions, and they are in direct touch of the antigens. They're directly touching the antigens and, you know, sensing the structure of the antigen, because we know antigen is also a protein. Antibodies and antigens both are the protein structure, but antigen is the protein part of the microbe, right? And antibody is your body's part, right? Is your immune system part. So when this hypervariable region is in direct touch of the antigen, it gives more fitted region for that antigen so that it can fit properly. So all in all, this hypervariable region is helping the antigen to fit as precise as possible, right? So variable region is clear, hypervariable region, then constant region, this FC and FAB region is clear, right? And also hinge region. Hinge region is that site, is the region from where Papin breaks down the antibodies. And when Papin cleaves or breaks the antibodies, it divides into 3 fragments, right? So this is the basic overview of the antibody structure, which is quite important for you all to understand, right? Now, very important facts I have to tell you regarding antibodies. This is the basic structure of antibodies. You all must be clear with this diagram, right? Now, there are total 5 types of antibodies we have, right? So I'm just telling you, and please listen carefully. There are 5 types of antibodies we have in our body: IgG, IgA, IgM, IgE, and IgD. Okay, in short, you can say gamed, just like we see the game. So you can add D in last, right? Gamed. So with this, you can remind all these antibodies. So what does it mean, this G, A, M, how we have like categorized their name, right? So this nomenclature has given to the antibodies depending upon their shapes, right? They all have basic structure is same, but they may have one or more heavy chains, right? For example, if I talk about IgM and IgA, they have one more constant region of the heavy chain. Okay, that means CH1, CH2, CH3. Basically, these three domains are present in the antibodies. Okay, but IgM and IgE have one more region of the constant heavy chains. Okay, so total 4 regions are there: CH1, CH2, CH3, and CH4. Now, IgG is the most abundant antibodies which is found in your body. Okay, IgG is the most abundant body which is found in your most abundant antibody which is found in your blood, right? And IgG means immunoglobulin, and this G means gamma. Immunoglobulin gamma, right? And IgA, A is for alpha, again immunoglobulin alpha, immunoglobulin, and then comes your M, right? M is for mu. You must have learned that in chemistry also. Then comes your IgE, E is for epsilon. Then IgD, D is for delta, right? So gamma, alpha, mu, epsilon, and delta, right? This is the basic expanded form of the antibodies, right? Now some other facts I'm telling you about the antibodies. We all know that antibodies present in our immune system, right? And the immune system is composed of some specific immune cells, right? Primary organs are there, secondary organs are there, primary lymphoid and secondary lymphoid organs, right? We know that basically the immune antibodies producing cells, B cells and T cells, they are manufactured. Where they are manufactured? T cells, maturation takes place in thymus, and B cells, maturation takes place in bone marrow, right? This you all are clear with, right? Now, after the formation of antibodies, they're released in the blood. So where does they reside? This is the question. In your body, or in fact in your blood, where antibodies are living, where they are present. This is important to understand. See, antibodies are present in your blood, but basically which component of blood is carrying the antibody? It's your plasma. Okay, now in plasma, in which component? So in plasma, we have two special proteins, globulin and albumin, right? So antibodies are present in the gamma chain of the globulin protein of the plasma of the blood, right? Again, I'm repeating, please listen. Your antibodies are present in blood. And which component of the blood is carrying the antibody? It's plasma. Which component of the plasma is carrying the antibody? It's the protein. And the name of that protein is globulin, right? And we know that proteins are made up of different types of chains, right? Peptide chains. So in which chains these antibodies are there? Gamma chain. That's why sometimes we also call it gamma antibodies. Okay. So now we know that why we call it immunoglobulin, because it is present in the globulin protein of the plasma of the blood, right? And why we call it gamma? Because it is present in the gamma chain of the globulin protein, right? Now, another fact which I have to tell you. See, we know that antibodies are divided into 5 types. Just now I have told you, right? IgG, IgA, IgM, IgE, and IgD. And the most abundant one is IgG, and the least abundant is IgD. Very few traces are there of IgD, okay? And also we do not know much about this IgD antibodies, right? So most abundant is IgG. The second most abundant, IgA, then IgM, then IgE, and IgD. The same sequence gamed, right? Now, you know, when you are present in your mother's womb, so she not only gives you nutrition and, you know, and takes your excreted waste out, she also gives you antibodies, right? It's quite beautiful maternal gift from your mother. Antibodies she gives you. She gives you the ability, ready-made ability to fight against the disease. Okay, so which antibodies your mother gives you? IgG. Okay, that means this IgG antibody can cross the placenta, right, and enters in you, right, in your cells, and through colostrum. Colostrum is that milk. Okay, so when mother feeds the baby, so you get IgA antibodies through the colostrum. So we can say IgG and IgG is gifted by your mother, right? Again, I'm repeating, IgG and IgA are the antibodies gifted by your mother to you, right? So this was all about antibodies, right? And about types of the antibodies we will learn in detail in next lecture. For this lecture, this is enough, right? I hope you all have understood and you all are clear with the structure of the antibodies. Thank you so much, everyone.